Hey guys, okay. This is the title of the research article I made a summary on. It's kind of long, but bear with me. I'm going to say it anyways. The involvement of the chemokine CCL3 in the prenoceptor P2X7 in the spinal cord and paclitaxel induced mechanical allodynia. So yes, it sounds very long, complicated, but it's a simple process really, and I'll explain everything to you. So, first off, the article was published on August 14th, 2015. Studies show that 38.5% of men and women in America will be diagnosed with cancer sometime in their lifetime. This is really sad because 38.5%, especially of America's population, is a huge amount of people. But luckily enough, there are treatments for this, such as chemotherapy. Chemotherapy is the usage of chemotherapeutic agents to treat tumors, one of which is paclitaxel. Paclitaxel usually treats tumors in breast, ovarian, and non-small lung cancer specifically. However, the usage of paclitaxel treatment causes neuropathy. Neuropathy is the damage of peripheral nerves, which causes symptoms of weakness, numbness, pain, and tingling in a person's hands and feet. Peripheral nerves are located in the central nervous system, and their function is to relay messages from the brain to parts of the body. So for example, if I want to say hello, for example, like hi, my brain is sending messages to my hand to move through the peripheral nerves. So the damage of the peripheral nerves can cause catastrophic effects on the body's movement. Also, it was recently discovered that paclitaxel treatment activates microglia. Microglia are primary immune cells in the CNS, specifically located in the spinal dorsal horn. So the main job of microglia is to detect inflammation in the body. Specifically in the central nervous system, either from an injury or the presence of a virus. What they do is, stimuli sent from the inflammation area travels to the microglia, activating them from the resting state. The microglia then travel to the inflammation area, and they phagocytose. Phagocytose is the ingestion of debris or cells. So they ingest, dead nerve cells and, pathogens, which helps reduce the inflammation there. And also in this process they release cytokines and chemokines, which contribute to the initiation of hypersensitivity. Hypersensitivity is unwanted reactions, from the function of the immune system. So let's say for example, allergies, but in this case it's way worse. Yeah. So as one of the cytokines they release, microglia produce CCL3. So, in case of a spinal nerve injury, the microglia release CCL3. CCL3 is responsible for the expression of and the response of the injury stimulus, which contributes to mechanical allodynia, which is just fancy terms for pain. So, however, there are receptors to inhibit the effects of CCL3. For example, CCR5 and CCR1. So the blockade of spinal CCL3 reduces pain and hypersensitivity after traumatic nerve injury. So these results suggest that spinal CCL3 has an important role in the traumatic nerve injury-induced allodynia. So CCL3 is responsible for the expression of pain in the body in the nervous system. So the aim of this lab was to investigate the specific role of CCL3 in mechanical allodynia. And by doing this, they used a rat model in which they simulated these conditions of using paclitaxel and neuropathy in rats. So a group of male rats were imported from Japan and were fed food and water and were injected with paclitaxel and a macroglia marker, which is a macroglia marker to mark the presence of macroglia over the course of 14 days. So they were injected on day 1 and on day 7. So I have like a little, like that's the guy from Ratatouille, Remy, and yep, it's big in Japan. It's a Alphaville reference. I suggest you look it up. It's really cool. Okay, so over the course of 7 days, the presence of macroglia was detected, and its concentration did increase. So this shows that macroglia was activated in the neuropathic for treated rat, in the paclitaxel treated rats. Also, 7 days after the first paclitaxel injection, the expression of CCL3 was also detected in the spinal dorsal horn. So, however, over time, like towards day 8 and day 9, the scientists examined the expression of CCR5 and CCR1, an increase in its concentration and the decrease of CCL3, and also the decrease of mechanical allodynia. So this shows that CCL3 was blockaded by its bondage to receptors CCR5 and CCR1, which was the cause of the decrease of mechanical allodynia. So here's a graph that shows the rate of the mechanical allodynia decreasing over the span of 7 days after the rats were injected with paclitaxel. So this indicates the neutralization of CCL3 with as it bonded to receptors CCR5 and CCR1, which caused the decrease of mechanical allodynia. So this study showed that an antibody to neutralize CCL3 reversed the established mechanical allodynia in the paclitaxel-treated rats. So therefore, the lab's findings produced evidence for a new method in the pathogenesis of chemotherapy-induced peripheral neuropathy. That's just fancy words for like saying like there's a new mechanism in the development of chemotherapy as time progresses. And also just to suggest a new therapeutic approach to end neuropathic pain. So Homer here is supporting that, and that's great. And then here, it's not the end, but a start of a new beginning. So I don't know.